So as of the last class, we left off with the citric acid cycle. We spoke about glycolysis and its role in cellular respiration, how it happens out in the cytoplasm, and the overall yield of that portion of cellular respiration is 2 ATP, 2 NADH, remember those are our high energy electron acceptors, and 2 molecules of pyruvic acid, also known as pyruvate. Now, again, as we talked about any type of enzymatic cycle, the products from the previous portion are going to serve as the substrates for the next. And so therefore the 2 pyruvate molecules are going to go into the citric acid cycle. Alright, so as we said, glycolysis, received the glucose molecule, broke that in 2, released electrons, ultimately forming 2 pyruvate molecules, and now we're in citric acid cycle. This is also known as the Krebs cycle, and this occurs in the matrix of the mitochondria. Be certain to review the structure of the mitochondrion with its inner and outer membranes, the inner membrane space, the cristae, the matrix, etc. So the matrix, we know, is the fluid-like material on the inside. And so the citric acid cycle, as I said previously, has 2 particular steps. It has the intermediate phase as well as the Krebs cycle proper. It begins with the intermediate phase, and this phase is basically the grooming portion that's going to take that pyruvic acid from glycolysis, and it's going to chop off one of the 3 carbons that are on those 2 3-carbon chains and make them 2 2-carbon molecules known as acetic acid. And that carbon that is taken off is released in the form of carbon dioxide. So that is a byproduct of the Krebs cycle. And so from doing this, from taking off of these carbons, we also release 2 NADH molecules as well. Now, from here, the acetic acid, before it goes into the Krebs cycle proper, attaches to what is known as coenzyme A. I say that this is like going to the prom. The person gets dressed up for the prom, and now they have to go find their date. And so coenzyme A is the date, and when it binds with this, forms acetyl coenzyme A, or acetyl-CoA. And this acts as the escort, is going to take each one of those acetic acid molecules through the Krebs cycle proper. So basically we have 2 acetic acid molecules from those 2 pyruvates, and so therefore we're going to go through the Krebs cycle proper 2 times. Alright, and then after each one of those molecules goes through this Krebs cycle proper, the CoA, or the coenzyme A, is going to be released and recycled for future processes. So again, as we see here, the pyruvic acid, we take off carbons, making them 2 carbon molecules instead of 3. The NAD plus comes in and picks up any excess electrons that are going to be released from that grooming process to make NADH. And then the coenzyme A is going to come in and bond with it to release in an output of acetyl-CoA. And as we see here, we show that the acetyl-CoA goes in and the Krebs cycle turns twice. Now, as a yield from the Krebs cycle, we receive 3 NADH in addition to those that we received previously with the intermediate phase. So each turn, so we're going to end up with a total of 6 NADH, 1 FADH2 for each turn, so a total of 2 FADH2s, and 1 ATP molecule for a total of 2 of those. So total production is going to be, we're going to get a net gain of 2 ATPs, right, 1 per turn of the Krebs cycle. We're going to get a total of 8 NADH, 2 from the intermediate, 6 from the Krebs cycle proper, and we're going to get 2 FADH2s from the Krebs cycle proper. And that leads us to the end part, which is the electron transport chain. Now, the electron transport chain, as we mentioned before, this is basically the bank. This is where we're going to get the majority of all of our ATP. Because if you think back from glycolysis to the end of the Krebs cycle, how much ATP did we form overall? All right, we only formed 4 molecules, yes? 4 were formed in glycolysis, but only 2 were a net gain. 2 were utilized to run glycolysis itself. So 2 from glycolysis, 2 from the Krebs cycle proper, and so therefore we only have 4. Well, we said that we typically have a yield of about 32 to 38. And so if we're going with the 38 aspect, the majority of this, regardless of what number, is going to have to come from the electron transport chain. Okay, so again, glucose, glycolysis, pyruvate to citric acid cycle. What do you think is going to come from the citric acid cycle into the electron transport chain? What is formed? We know that acetic acid was made from the pyruvic acid, but then that combines with the coenzyme A, and then that's broken down, so the coenzyme A is then recycled after its release. So the only thing left would be our buddies NADH and FADH2. Again, we said that they're the ones that are picking up the pennies, picking up those electrons. And so it's that money that we're trying to take to the bank so that we can get cash. Cold hard ATP. So basically, electron transport chain is going to occur in the inner mitochondrial membrane. Again, the ETC is known as what? Oxidative phosphorylation. Okay. Now the ETC basically it's a chemical machine. It's like a conveyor belt basically. There's a variety of things that are going to happen in order for this ATP to be produced. This is basically all about turning on the ATP machine. And that ATP machine is known as ATP synthase. So several things have to occur in order for the ATP synthase to turn on. And so first of all, we're going to have the electrons taken to the bank. Those electrons are going to be dropped off, and from that there's going to be resultant osmosis or chemiosmosis of hydrogen ions from the matrix region to the intermitochondrial space. Alright, these hydrogens are going to cause a buildup of potential energy, and from that potential energy, that is what's going to result in the ATP synthase turning on. Alright, so we're going to have some other key players in here. For example, the cytochrome molecules that are listed here. These cytochrome molecules are necessary for picking up the electrons after the NADH and the FADH2 drop them off. They're basically going to pass them along the membrane to one another. And as a result of that, water is also going to be formed. Okay? So, as the chemical machine gets ready to turn on, what's going to happen is that hydrogen that built up that potential energy across the membrane is basically going to release energy, that potential energy, and it's going to want to come back in through the special entrance known as ATP synthase. When it does that, it turns the machine on. That ATP is picking up all of that, ATP synthase is picking up all of that energy from that hydrogen flow, and it's going to synthesize ATP in the process. And how it does that is it takes inorganic phosphate and bonds it to ADP. Okay? Now there's a rule of thumb. How are we going to get these 34 molecules of ATP? Well, let's think about this. Let's really think about this. We take our money and we change it in on a regular basis, right? If we take our dollars to Canada and ask for a currency exchange, what's going to happen? Are we going to get the same amount? No, there's going to be a difference in the amount. And so what's happening here is NADH, which is our high energy electron acceptor, is going to be worth more. It has greater value. And it has a value of 3 ATP for every NADH that steps up to a cytochrome, steps up to the ETC. For FADH2, it's going to have a worth of 2 ATP. Okay? And so as we see here, basically, the NADH and the FADH2 are going to come up to the cytochromes and they're going to dump their electrons. Alright? Those electrons are going to be passed from cytochrome to cytochrome to cytochrome, these proteins that are embedded in the inner membrane of the mitochondria. Alright? As a result of that, as we see, we're going to get a formation over by the penny. We're going to take oxygen and we're going to add it to those electrons that reach that cytochrome, and as well as some hydrogen molecules to form H2O. Also, we're going to stimulate what is known as chemiosmosis, the movement of those hydrogen molecules that are inside of the mitochondria in the matrix out to what is known as the inner membrane space, and this is going to build up potential energy. These hydrogens, as they build up potential energy, are going to want to come back into the mitochondria, and how they do this is not the same as how they left, but they're going to come in through what is known as the ATP synthase. And as they enter through the ATP synthase, they turn it on, resulting in ADP binding with phosphate. Okay? And lastly, one of the things that's going to stop this whole process is a molecule known as cyanide. Again, it's an inhibitor. It's going to stop the chemical process from occurring, but in very tiny amounts. That's why when people get cyanide poisoning, that cyanide poisoning is stopping ATP. Okay? Alright, so again, as a summary of everything, we get 2 ATP from glycolysis, 2 ATP from the citric acid cycle, and 34 from oxidative phosphorylation, or the ETC chain. Alright? Okay. So if you study this slide, basically, this is summing everything up, or if you study the table that we've talked about, you should be able to process everything and understand what the overall yields are for each of the steps involved in cellular respiration. And so when we look at cellular respiration, we primarily only talked about glucose. But also remember, if you're not eating, your body still has to make ATP, and so therefore our body has to be able to take other types of things to create ATP. Okay? For example, other types of carbohydrates can be utilized. Fats can be utilized. Again, long-term energy storage, right? And as well as proteins. Proteins are very important for ATP synthesis. And so if we look at this, they're all going to have a different type of processing. Right? Some might be somewhat similar, but others are going to be totally different. So if we look at it, polysaccharides, okay, they're broken down into sugars, right? Polysaccharides are those that have multiple sugars. And so all these sugars are going to go directly into glycolysis. If we look at fats, when broken down, looking at their structure, they have a glycerol molecule and they have fatty acids. Okay? Those glycerol molecules are going to enter directly into glycolysis, just like glucose. But the fatty acids are going to start at the intermediate phase of the Krebs cycle. And then if we look at proteins, we know that proteins are constituted of amino acids. And those amino acids, when you break them down into individual sections, breaking down their structure, again, the carboxylic acid, the amino group, the functional group, etc. Each portion is going to have to enter into the cellular respiration at different places. For example, one portion might enter in glycolysis, another may enter into the intermediate phase, and then another in the citric acid cycle, all of which then go to electron transport to yield ATP. Now, what happens when we don't have oxygen? In the case of fermentation or anaerobic respiration. For example, some of our cells, for those short twitch muscles, as we talked about with the sprinters, they can work for very short periods without any form of oxygen supplement whatsoever. Also, various types of organisms can utilize that whole fermentation to harvest their food energy. Mussels function about 15 seconds, basically, before they kick into the whole anaerobic respiration process. And so how fermentation makes ATP is that it relies primarily on glycolysis. Some of the other steps basically are going to be completely eliminated. And so without that oxygen, we also had to remember, excuse me, we also have to remember that those ATP molecules that are going to be made basically aren't going to be as great in number either. And so we're only going to get a yield overall of about 2 ATP from fermentation in comparison to the 32 to 38 from cellular respiration. And so for each glucose molecule, basically what we're doing is we're doing the same thing. We're breaking it down to pyruvic acid. But it doesn't end there. That pyruvic acid, before it exits glycolysis, is going to be converted into another product. All right, and as it's converted into whatever product that may be, lactic acid, carbon dioxide, etc., we're going to have the NADs picking up the electrons from the initial conversion, but that conversion from pyruvic to whatever the end product is going to require those electrons back, and so that NAD plus is going to end up being the end result. So it's going to go in as NAD, picking up the electrons to make NADH in the conversion of glucose or whatever it is to pyruvic acid, but then pyruvic acid to whatever that end product is, that NADH is going to come back, donate the electrons, and then leave as NAD+. So basically the whole glycolysis process is going to continue. For example, in human cells, we make lactic acid as an end product of fermentation. And so we start off with the glucose. The NAD goes in, picks up the electrons, making NADH. Then we have two pyruvic acid molecules, and then those two molecules must be subjected to another conversion, and so those NADH come back in addition to hydrogen, and we get NAD and lactic acid. We've talked before, various microorganisms utilize this process of fermentation to keep up their energy levels. Sometimes lactic acid is the end product, for example in dairy products, such as cheese and sour cream and yogurt, but then there are also soy sauce and pickles and olives and meat products. The list can go on and on and on. Yeast does the whole alcoholic fermentation process. It gives off carbon dioxide and ethanol or ethyl alcohol instead of lactic acid, and so that's how we get a lot of the alcohols that we drink. That's how we make bread. Yeast goes into the whole bread fermentation process. You put yeast, you put sugar, and the yeast feed the yeast feed off of the sugar, and so you get that alcoholic fermentation and that carbon dioxide produced. But again, we don't get drunk off of bread primarily because when we bake it, it comes off. And so as you see in this particular table, we are seeing a variety of different organisms and how they handle anaerobic respiration. So we go from glucose to pyruvic acid, and then now we have to take that pyruvic acid and convert it into something else. What did we see? Propionibacterium basically is going to have an end product of CO2 and propionic acid, and as a result of this, we have the formation of Swiss cheese from milk. Aspergillus, Lactobacillus, Streptococcus, Lactobacillus acidophilus is the one that you hear about all the time. Live and active cultures and things such as yogurt, sour cream, cottage cheese, soy sauce, cheddar cheese, again, they produce lactic acid. Saccharomyces or yeast, that's the technical name, Saccharomyces. Again, alcoholic fermentation, making ethanol and carbon dioxide so that we can get things like wine and beer. Clostridium. Okay, Clostridium is the one that is responsible for making acetone and isopropanol, giving us our nail polish remover and our rubbing alcohol. And then we have our E. coli, our Escherichia, and our Acetobacter, making byproducts of acetic acid, giving us our vinegar. Alright, and so these videos are available online in Blackboard so that you can watch some tutorials on the whole cellular respiration process. Again, remember, if you have any questions, please be sure to contact me. And I hope you have fun studying.